Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you. Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. Make sure to check out our newest book on understanding viruses. This book is a compilation of interviews of 25 plus top virologists, and the questions I asked made pretty much all of them say, that's a good question. So there's a lot of speculation, a lot of new information, I believe, that will keep people thinking and experimenting for decades to come. So if you want to get the book, it's not expensive. Go to Amazon.com and type in Finding Genius. So my guest today is Yanlan Mao. She's a group leader at the MRC Laboratory for Molecular Cell Biology at University College of London. So we're going to go into her work. So Yanlan, thank you for coming.
Thank you for having me. It's a pleasure to be here.
Well, tell me about your research, please.
Yeah, sure. So overall, I'm basically interested in how tissue size and shape is controlled and how we can get this beautiful diversity of animal and plant sizes and shapes in nature, even though actually we're all, at the very basic level built of very similar building blocks. So specifically, my lab is investigating this from 3 aspects. One is, how do tissues actually get to their right shape as they're growing and developing? Another is how do they stay in this shape during the lifetime of the organism, which is often the longest time period they have to cope with. So how do they actually stay in this shape despite the constant fluctuations and perturbations that the environment may be exposing the organism to? And then finally, if they are injured, for example, how do they actually remember their original shape? How do they get back into their correct shape after injury? So basically wound healing or tissue repair processes.
In embryogenesis, I've wondered, let's say there's 120 billion people that have ever lived. Why do they all, not all of them, but almost all of them seem to have one liver, not 2? And why is the liver on the right side if they look down and not the left side? And why is the pancreas in opposition? And why are there these shapes? And, what governs all of this? So I'm sure you're looking at the same question.
Yeah. For example, how is the heart shaped? How does it generate shape, its morphology, and why is it always, as you said, in different positions? So this is all to do with kind of positional information, tissue morphology at the individual organ level, but also at the kind of whole system, whole animal level. How are these different organs coordinated? How do they position themselves, as you say, correctly in order to presumably carry out coordinate functions, right, at the whole animal level? So I study, to be honest, less at this kind of animal level coordination. What I look at is more at individual organs. So for example, we use the fruit fly Drosophila as our model tissue and the wing specifically as the actual tissue. And so we use the wing as a model tissue to understand, how does the wing get to its correct size, correct shape? Because obviously it's harder to, look into that in humans, although people are starting to look at that now using different methods. But so what we know is that, and what we're interested in is, for example, of course, all the information is in our genes. We look like our parents, our kids look like us, but exactly, how do these genes, even if we understand, the genes and the sequences, how do they actually, generate the proteins which then drive morphogenesis? And to understand that, we really need to understand forces. So, because ultimately to sculpt any tissue or any object, any matter, you have to apply a force to the object, to the matter, to make it move, to make it, change shape. And so it's really a very physical process, a mechanical process. And that's really what my lab is interested in. How do mechanical forces affect cells, how they behave, how they change shape, they grow, how they divide, and also how they repair themselves if there is injury. And so we use, in order to help us understand these questions, we, offer experiments, so on. Importantly, a very important tool in our lab is to use mathematics to understand these in a more kind of complete, holistic way.
So what?
Did you hear me there? Sorry, my internet said it was unstable.
Yeah. So what are the factors in terms of organ morphology? Again, embryogenesis or gestation is a very active time. Then there's some creatures that can regenerate lost limbs, salamanders, things like that. And I guess, cancer is probably different. But what are the morphogenetic events that are important to you in an organism's life? Yeah,
so there are many. So one hugely important process is really the process of cell proliferation, cell division. After all, most of at the very beginning we're all single cells, and so in order to create complex organs, complex shape or multicellular systems, is the first thing morphogenesis is really how do you grow and then divide your cells, right? And just controlling proliferation rates, these growth and division rates, is already a very important process to get correct. And you mentioned cancer, because of course, when these things aren't correctly controlled, you get diseases like cancer, where it's basically divisions are out of control, and the whole structure loses its shape. But it's not just dividing as a kind of a ball of matter, because again, at the beginning most embryos are spherical, but very quickly they start to adopt different shapes, right? For example, it goes from a sphere to an ellipsoid, so you already have axis elongation, and there's already processes there. So for example, one process we looked at in the lab before is the process of orientation of divisions. How do you control how to divide a cell in an oriented way? And what we found is that actually mechanical forces is very important in that. So what is being shown for almost over 100 years ago now is that cells when they are elongated, they tend to always divide to bisect the long axis, so that a long cell gives rise to two daughter cells that are more spherical, which makes sense. But the cell shape is actually very influenced by mechanical forces, whether it's intrinsic forces in neighboring cells stretching these cells, or the cell growth and the material properties of the cells are isotropic. So for example, if the cell has stiffer ends, then like an elongated balloon, when it's growing, it won't grow as a sphere, it will grow as a sausage shape. And then when it's about to divide, it will orient its divisions in this, along this elongation axis. So that's another key step, orientation of divisions. But also another very important process, and for example, just the simple process of elongation of a whole tissue, is what was known as cell intercalations. So for example, cells, they can stick together, but they can also change neighbors. So, let's say between a group of 10 cells, if we start to rearrange our cells, we could reshuffle and shuffle our position, so that instead of, let's say 10 by 10 row, I end up being, 20 by 5 row of cells, right? Then you can see how that geometry is really changed from sort of spherical isometric shape to an elongated shape.
What do you think is the cell-to-cell signaling that governs morphology? Like, again, if we look at it in a liver can regrow, right? Well, have you or anyone observed when a liver is lacerated or part of it's resected, what does the regrowth look like, and what does that tell you about the signaling that could be going on?
Yeah, so I have to say I'm not a liver expert, although I do have a colleague, and we're actually starting to work together to look a little bit at liver regeneration. So I know a little bit about liver. So for example, in terms of the liver, depending on the size of the cut, whether it's a small cut or a large cut, you obviously damage different cell types. So the liver has, different cell types such as hepatocytes or the ductal cells, the cholangiocytes and so on. And so from what I understand is that if it's a small cut, generally the hepatocytes will just, proliferate and heal that small missing part of the tissue.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
But if you have a very big kind of, huge cut, let's say almost half of the liver is gone, and amazingly, the liver will be able to regrow even, if half of it is removed. And then what happens is these cells have to de-differentiate. So this is, not a mechanical problem, although we are in this new project trying to understand the mechanics of potentially this behavior. And I can tell you, is often these hepatocytes, they will de-differentiate to become what's known as a more progenitor-like state, so that they're more stem cell-like in a way. And then these cells will proliferate better because when they're more progenitor-like, they can divide more. So again, going back to, the earlier question, the first thing is they have to build up their cell number, right? They have to increase cell number dramatically. And then when they sort of have overcome that lost cell mass, then they start to differentiate again into whether it's hepatocytes or cholangiocytes or ductal cells, and start morphogenesis, for example, forming the bile canaliculi so that they can undergo the correct functions, bile formation, and so on. And so one interesting aspect, of course, is, it's being shown that the liver seems to have different mechanical properties. So, for example, the cells seem to have different tensile states, depending on whether it's in a more progenitor-like state or in a more kind of mature, hepatocyte-like state. And what we're looking into is how does the kind of mechanical properties of the cells, for example, how stiff they are or how much tension, they're pulling on each other, how can that actually affect its ability to regenerate? Because all it's being shown in disease conditions, right? A lot of liver disease, extremes, fibrosis, cirrhosis, so on, they that actually are less regenerative. And so that's why we're thinking perhaps, the stiffness of the cells and their ability to sort of apply tension to each other, to move, to induce, for example, cell proliferation. And a new concept that we sort of looked into in the lab is the concept of tissue fluidity. So the ability of cells to move and intercalate relative to each other. These could also all contribute to helping, how a tissue is able to repair itself. I mean, I'm not saying this is being all shown in liver. What I'm saying is these are concepts that, are sort of known in other tissues and other animal species, but potentially they may also work in the liver because you were, raising liver as a sort of interesting organ.
Has anyone observed, let's say, just cell division, either in bacteria or somatic cells, and, every few seconds, let's say, query the tensile strength of the cell membrane? Can you do such a thing? And if so, what's been observed over a period of time? Or again, in embryogenesis, I don't know, for a mouse or whatever model you can use, or Drosophila, can you evaluate that?
Yeah, absolutely. So in different, model systems, whether it's cell culture, obviously it's been easy in cell culture. So for example, people have used like HeLa cells and measured, for example, the forces the cells can generate when it's, dividing under mitosis. Often there's a drastic cell rounding. So that itself, cell division itself is a huge, mechanical process, morphological process, because the cells have to change shape, they have to get round, they have to get bigger for the apparatus to, for the cell division apparatus, to build. And so yes, people have measured, forces that, a cell can exert during these different times. And most of being in cell culture, including, cancer cells, wild type cell lines. And often it's being shown that cancer cell lines are actually able to resist, and push harder so that they can sort of grow and divide more and better than its neighbors in the compressed kind of environment. But also in for example, the Drosophila wing disc, which we've been, looking at, it's a more complex, in vivo system, of course, it's multicellular tissue. You could argue that's more, physiological, more like how real organisms grow and develop. And in those systems, we've been, for example, to carry out live imaging where we can, image the tissue grow and divide from very small stage, only sort of a few hundred cells and up to, 100,000 cells or so. And, we can track that process.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
For a few hours at a time to look at how the cells are growing and dividing. And we've been able to perform experiments such as laser ablation, which is basically you use the laser like a pair of scissors and you can cut on the cell membranes or the cell kind of junctions and use this to evaluate the tensile forces in the tissue. So the idea there is imagine the cells are all connected like a network, like a web. And if you use scissors to cut this web, if the web is under a lot of tension, then you will see the web recoiling, right, bouncing back. And so we can use these sort of very high-powered lasers, but very sharply controlled localization, very precise localization of these lasers to make cuts in cells at different times. For example, when it's dividing or when it's growing or when it's undergoing, different types of cell movements to understand what the forces are at the kind of cell membrane level.
What makes the forces arise, though? What governs the tensegrity of, the cell membrane in a particular place? What modulates it?
Yeah, great question. So there's a whole class of molecules collectively known as the cytoskeleton. So in the simplest term, for example, what's kind of dominant in most cells is this network known as actin-myosin. So actin is basically kind of a mesh of polymers. And when I say mesh, basically actin can organize into different types of structures. And these tertiary structures will dictate often the shape of the cell. But another molecule known as non-muscle myosin 2, so a bit like, muscle myosin that we all know of, these myosins are motor molecules. They can cause contraction of these actin fibers sliding against each other, a bit like, muscles are able to slide and contract. So imagine that kind of a network, but around cells. So basically you have this kind of, think of it as like a meshwork that's able to contract and expand depending on how many molecules of myosin you have and where the myosin is localized. So for example, if the myosin is more of it on the right half of a cell, then this half of the cell will contract more, right? And the cell will be small on one side.
Is the myosin captivated by particular parts of the cell? Is it mobile? Or what is it attached to? Does it stay in the same place?
Yeah, so the myosin is mobile. So there are also cytoplasmic sort of pools of myosin that can flow around the cell. But what attaches these is basically the actin binding. So the binding of the actin, the affinity of the myosin to the actin will cause it to, first of all, to localize to the actin. But then also there are other molecules upstream that will control, for example, the activity of the myosin, how much phosphorylation it is. There's other kind of regulatory chains of the myosin that will activate it, which will cause motion and then contraction. So absolutely. So there's a lot of signaling pathways and sort of biochemical signaling pathways that will lead to the downstream activation of this myosin. And as a result, this will generate forces in different cells, in different locations at different times. So then you can imagine this control will, give you really dynamic kind of emergent behavior of the whole tissue, of the whole system, basically.
Has anyone, I don't know if this probably be very hard to do. Can you put in fluorescent dye markers or some kind of marker and then observe, in vivo in a live cell, the structure of the interior of the cell changing? The migration of myosin, the stiffening and relaxing, and maybe even the porting of various chemicals throughout different parts of the cell. I would think that this physical network pushes and pulls and moves all kinds of materials throughout the cell.
Yes, absolutely. And that's something that's really, technology has changed so much over the last even just 5 years. We are able to either through dyes or through generating fluorescent proteins. So tagging, for example, GFPs, RFPs, different color fluorescent proteins onto different molecules of proteins of interest. For example, we could, tag the myosin with GFP, which we use in the lab. And at the same time, tag actin with, red fluorescent marker. And simultaneously, we have a blue fluorescent protein that we can use to mark, another whatever molecule of interest that we're looking at, at the same time. And then we can then image this whole cell at very high, subcellular res as it's growing, as it's dividing within a whole tissue even, or, in a single cell kind of context, in a dish. But what's challenging now is really trying to understand subcellular detail. For example, as you said, how do these different, pools of actin myosin, how do they move dynamically? How do they change shape of the cell while the whole cell is still embedded within the whole complex tissue where there are hundreds and thousands of other cells? And so exactly, technology is really amazing now, allowing us to use different types of dyes or fluorescent protein technology, where we can then image a lot of these processes all together in its physiological tissue context. And that's super exciting right now.
Well, what's been observed so far? Like, again, has anyone done a study of a cell dividing or, tensegrity changing in a tissue over time as it, the liver before and after a meal, it changing and moving? Like, in what modeling have you been able to do, and what's been observed so far?
Yeah, so there's, lots and lots observed so far. So, for example, I can, give you a little story that we looked at in our lab with regards to combining computation modeling and imaging.
Okay.
Observe that a different, what we were interested in is basically, what polarity of forces, how do polarity of forces within a cell, how is that able to orient cell divisions, which we know actually gives rise to, for example, the typical elongated shape of the wing. So if you look at most insect wings, they have a long axis and a short axis, right? So our question then was, how does the wing get this shape? And we know it's because the cell divisions are oriented in a particular way. So then we asked, well, how do these divisions orient themselves, right? And so then, cut long story short, we were able to observe through imaging, for example, tagging fluorescent myosin, able to show that the myosin actually is polarized. By that, I mean the myosin is not everywhere around the cell, but it's only on sort of two sides of the cell. And so as a result, these two sides of the cell, they have different tensile forces compared to the other cells. And this is what gives rise to the cell's anisotropy. So then when it grows, it doesn't grow kind of into a circle, but it grows into oval shape and elongated shape. And then when the cell then divides, it follows the long shape axis rule and divides in an elongated way. And also as a result, we were able to show that computationally. So when we were looking at this, using a model, so a lot of people were skeptical. So, well, how do you know this myosin, this atypical myosin called DAX, how do you know it's not directly, for example, doing something else? It's not to do with forces, perhaps it's binding to spindles and, tethering, completely different kind of cell division mechanism, right? And so the power of the computational models and mathematical models is that you can really control what you put in. Because after all, the whole tissue, the whole wing, all of these other factors could be playing a role. And you can never be sure, it's only the forces that's driving this behavior, right? And not something else these molecules could be doing. And of course, many molecules have multiple functions. And so it is very hard to say it never does something else, right? Because, it's very hard to prove it doesn't do something in reality. So the kind of mathematical models that we build, the importance of them is then we can use the models. For example, if in the model we only had forces and we didn't have anything else, right? And the models were able to explain most of the phenotypes we see in vivo, then we can say actually, in this case, for example, the polarity of these mechanical forces actually is a really dominant driving force in the behavior that we observe, which is in this case helping the wing orient its divisions and therefore control its size and shape.
Well, have you tried to make organoids and then put them in culture in a constrained mold of, let's say, a strange shape and see what happens? What kind of shapes do they make, the cells?
Yeah, so we haven't yet ourselves, although we are starting to use sort of organoids or 3D spheroids as such and change its mechanical environment, not just the shape, but also the kind of the stiffness of the gels that it grows in order to see if we can control the kind of morphology of the whole organoid, and its future behaviors. But others have done that in some different sort of complexities, whether it's in 3D or even just 2D micropatterning, where you're plating cells on a 2D kind of constrained or confined space, and then look at how the cells grow and so on. But I think as you said, is of course much more interesting in 3D. So just Lutov's lab, what they've done is started to grow intestinal organoids in sort of different confined spaces. So for example, they sort of 3D micropattern gels that already have the curvature of the gut villi and so on, and then they plate the cells on and let it grow. And of course, from its behaves much more like a mini gut than a blob of cells in the dish. And so again, I think with this type of technology, one can start to ask much more interesting questions like these. How does the shape of the environment, but not just the shape. After all, I think the environment is more than just a scaffold. It's more than just a barrier or a shell, but it can have much more kind of long-term, interesting consequences. So for example, if you had the barrier temporarily and then you took it away, what happens then? Do the cells just kind of bulge out again, or do they somehow remember that past, that history, that kind of mechanical compression or confinement that they had before? These questions, I think, become much more interesting than just, growing a square organoid, for example.
Right. Yeah. No, I agree. So what experimentation is happening right now? What specifically are you trying out to figure out?
So one thing we're following up very much is regarding tissue fluidity. And so what I mean by tissue fluidity is basically the ability of cells to move relative to each other. So normally, if you think of the solid tissue, the cells are always pretty much stuck to their neighbors, and there's not much movement, at least during adult life. But occasionally, sometimes what we found is that actually upon injury, so if there's a wound or a lesion, then the cells around the wound start to move relative to each other and become what we call more fluid. So think of, it's like water, therefore these cells are able to flow into the gap, to seal the gap. And I think this makes a lot of sense, because basically, if you're liquid, you take on the shape of your container, the space that you're in. And so if there's a hole because of the wind, then these cells literally flow into the hole to seal up. So this was actually a new kind of observation that we made with respect to wound healing, where this concept of tissue fluidity, which was not new, but applied to wound healing, what is new that we discovered a few years ago. And so we're very much excited by this kind of mechanism, by this principle. And so that was found in the wing disc in Drosophila, but now we're trying to see if this principle is also true in other tissues. For example, you mentioned the liver, and also, let's say, the kind of the different organoids, different mammalian epithelia, and such as the lungs and so on, where often there are mechanical lesions and damages. And so we're very much excited by sort of different experiments where we can test this concept of tissue fluidity. If we make a hole in the liver organoid, do the cells actually start to move more relative to each other to flow into the wound? And how do they actually control this, if it is true?
In Drosophila, is it possible to look at what's happening gestationally? Like, how long is their gestation? And, even in vitro, can you make an artificial, Drosophila, or environment for the cells to differentiate and grow that approximates what actually happened?
So I guess
flies lay eggs, right? So they're, I guess you have to recreate the egg.
The fly, the whole life cycle, is only 10 days. So, and also the embryo itself is already very amenable to, for example, imaging and manipulations and so on. So I guess there's not much of a need to try to artificially make it, because it's already so good at doing it itself, and we can access it quite well and image it quite well. So you could say that maybe this, although apparently actually I think there are some groups that are trying to make a synthetic fly embryo, see, because I guess
if you could tag, the first, I guess flies have zygotes, but the initial zygote, the initial cell that would make up the whole fly, and then watch it through gestation and see where the original cell goes. Does it die off pretty quick? Does it become part of the body?
Yeah. People are able to do that already. So we let's say labs, many labs such as Eric V. Schell's lab and so on are and because the imaging is so powerful now, you can really do total imaging of the whole initial embryo, so the fly embryo is kind of like a syncytium. So it's a single cell at the beginning, but then there's lots of cell divisions that will happen in it. There's cellularization. And so people have made some amazing movies to really understand these early stages, and then actually be able to image the whole kind of embryogenesis process. And it's which takes about a day or so. So it's in terms of its kind of time scale and the size of the.
Have you seen these? And if so, what does it tell you? Like, what interesting things have you seen? Have you looked at one of these, home fly movies?
Yeah. I don't want to sort of pretend that I'm an expert on fly embryogenesis because I'm not. But many labs are looking at this, and there are some amazing movies, on YouTube, and of course, in many published.
But again, with your specific eye and your specific experimentation, what do you see? Because you're thinking about morphogenesis all the time, So
yeah. So for example,
but what do you see? That's interesting. What jumps out at you when you look?
Yeah. So maybe not at me, but even when I look at it, but again, people who've looked in this field. In fact, the fly embryo has been a great model to look at tissue mechanics and morphogenesis, because of the fact that it's so easy to image these cells. The cells are very big, and using the fluorescent markers, we mentioned earlier, people have seen, for example, the cells, individual cells, how they can exchange neighbors. So there's this process called germ band extension, where the process of cell intercalation, which is neighborhood exchanges that I discussed earlier in this kind of tissue fluidity concept, where really the kind of molecular and physical mechanisms of how cells exchange neighbors, undergo these exchanges and elongate the whole kind of tissue axis. All of that work was actually understood using the fly embryo as a model, because again, let's say the first time anyone looked at these movies and saw these processes happening, you're like, just it's incredible. Wow, you see these cells change shape. There are certain junctions that shrink more than other junctions, and then they notice there's, for example, this myosin 2 that's localized just at those shrinking junctions, and so they were able to conclude that this myosin is really required for the contraction of these shortened junctions. But then these junctions are only shortening in, for example, the vertical junctions and not the horizontal junctions. So then as a result, everything is polarized. So you have collective shortening in one axis and then elongation in another axis. All of these. In fact, I think in our field, what we often do is basically when we don't know anything yet, we make a movie. we just see what we can see. Seeing is believing, and then by detailed analysis of this movie, whether it's the cell shape changes, the cell size changes, the cell movements, all of these morphometrics that we call, from that, often that's, the most revealing. And really that starts us sort of generating different hypotheses regarding what's happening. And then we can zoom in further. Then we can start saying, well, what are the molecules required? Are there any molecules? In order for this to happen, these, cells or these junctions must have higher tension than these ones. If so, we can measure this tensile forces using, for example, laser ablation. But even then, we can ask, well, what molecule is responsible? Then we can start, looking through the tagged library. So, for example, again, flies is great because almost all the genes are tagged with GFP now. And so we can screen, hundreds and thousands of different fly lines to look for the gene that's exactly localized in the pattern we think it should be in order to carry these forces, for example. And that's often how new mechanisms are discovered. And that's what the fly is great for, because you can really carry out sort of unbiased, open-minded genetic screens, for example. If you really didn't have a clue at the beginning, how anything is working, then you can really make these observations and screens and so on.
Okay. Are there any particular clinical outcomes that you think your study would be useful for?
So yes, absolutely. So for example, the wound healing stuff we're saying, so this concept of tissue fluidization. So we were able to show, at least genetically, we were able to actually make, again, the wing disc for now, but we're testing in mammalian tissues. We were able to genetically make the tissue more fluid. And as a result, the wound in the fly was able to heal twice the speed of a normal wound, which we think is pretty incredible. So now we're trying to test, sort of molecular compounds or, other, drug targets and so on that would also have the same effect in different epithelia, of course, more relevant to human health. So different mammalian epithelia, human epithelia, not just the skin,
why do you think
the lung, the gut.
Why do you think that the fly's wound healed so much faster? What was it that you think did it? Like, how did the cells move differently versus the way they normally move? Yes,
exactly. So basically, we know exactly what happens. So the wild type wing, is pretty good at doing it. It takes about 2 to 3 hours, depending on the size of the wound. But what we did is we knocked down genetically this myosin regulator, basically this myosin that we were talking about earlier, which normally sets the tensile state of the tissue, we reduced that slightly in the tissue so that it's not as tense. So imagine if you have, again, this sort of network web analogy. If all the cells are pulling on each other really tough, I'm pulling onto you, you're pulling onto me, we're not letting go of each other, then we're basically like a solid tissue, right? So we knock down this activity slightly so that sometimes I can give way to you, sometimes you can give way to me, and as a result the cells are jiggling more, they're much more fluid, and we can see that happening. So the cells, they don't fall apart yet, we haven't knocked down this sort of key protein that much, but we've loosened the tight web, is more jolty, if all the cells are moving and wobbling a bit more, and basically we reduce the tension in the tissue. So that's the key message. We reduce the tension, and as a result we made the tissue more fluid. And then when we make the wound is able to heal in half the time.
Well, have you tried to turn the dial to be very loose versus only a little bit loose? And if so, what do you observe?
Yeah, so it's sort of hard. It's actually hard to do that, at a very quantitative kind of level. So we did have some where we went too extreme. So we sort of loosened it too much and the cells basically just died. The tissue just kind of flopped because, again, this molecule is required for just the basic, tensile structure of the tissue. And also, again, if we knock down the activity very slightly, there wasn't really much an effect. And we couldn't really get a kind of a statistically significant change. So there is kind of a fine window. But I think the system genetically, therefore, is just not perfect for looking at these, because again, even every fly has its own kind of noisy, even wild type of a noise within, how well it heals. And so we thought maybe if we can go to kind of a more quantitative system, perhaps, compounds and drugs and so on, they may help us understand things at a more quantitative level. And if we can be more targeted in, where we affect these pathways upstream that are affecting this kind of tensile state of the tissue, then we can perhaps, have more control, which of course will be important and necessary if we want to think of this as a therapy, whether, how much you knock down, how much, for example, in terms of a drug or a cream you apply to wound site. Of course, it will be important exactly how much you apply, the quantity, the area of application, all of these will be very important. And so one has to really look at this in a much more quantifiable way, as you said. And I think, which is why basically we're starting to use, human epithelia that are relevant to kind of the wound healing model and screen for different drug compounds that may be important for that. But we're very early stages. However, I do think there is very much, a huge potential in that.
Okay. Well, very good. Yanlan, what's the best way for people to find out more about your work? Where can they go?
So Google my name and hopefully most of my papers will be freely accessible. So I've made sure they are open access, but also bioRxiv. Pretty much all the papers go onto bioRxiv first. And also feel free to email me, look at my website, email me, follow me on Twitter at Yanlan Mao, and or just drop me an email.
Okay, well, very good. Well, Yanlan, thank you so much for coming. I appreciate it.
You're welcome. Pleasure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.